Hi everyone, welcome back to another video. We are very excited today because we have a Laur from Plants in Jars today.
Hi guys.
And today we are going to be working with carnivorous plants. The plants where I got started, it was carnivorous plants, and we are going to start working with some of these. So on her channel, we already did made media and the sterilization for the Venus flytraps. Here on this channel, we are going to be working with Nepenthes. So we are going to start by making media, and then we want to show you how to sterilize the plant. And I also have some seeds that I'm going to show you how to sterilize those. And we're going to place those all in tissue culture, all in this video. If you want to see how to do Venus flytraps, make sure to check Laur's channel, where we're going to be doing the same thing, but with the Venus flytraps. So let's get started and let's make some media. So let's start by adding water to our media bottle. Let's start with 200 milliliters. So if you are here from Laur's channel, you saw that we make tissue culture media already. We are going to be doing the same thing, but it's going to be a slightly different because we are going to use the media that we use on her, that we made on her channel to place in the explants. However, for seeds, we need to make a slightly different media. So this media that we're going to make is going to be for the seeds. So let's start by adding the water. We're going to make 250 milliliters. So we start with around 200. And let's add the sugar. So all the numbers that we are going to be using are for 250 milliliters. If you are making 1 liter of media, just multiply everything times 4. So for sugar, we need 5 grams of sugar. Let's add the sugar directly to the bottle. So we have 5 grams of sugar. Next, we have our MS media. This is the media contains on the micros and macronutrients and the vitamins for the plants to grow. Now with carnivorous plants, we already mentioned on her channel that carnivorous plants, they grow in like a very poor nutrient soils or conditions. So if we use the full MS media, the plants will not going to like it and they're going to die. It's too much for them. So on her channel, we make media, we make 1/3 MS. However, for seeds, we are going to do a slightly different. We actually want to do less than that. We're going to do 1/4. So we're making 250 milliliters. So for our media, we need 0.37 grams.
Okay.
Yeah, so we need between 0.3 and 0.4 grams is perfectly fine. So we have the sugar, we have the media. Now let's add some ppm. This is a plant preservative mixture. This is going to help this is going to prevent the growth or contaminants. So it's going to inhibit the growth of fungus and bacteria that may be in the media. So for this, we are going to add the equivalent of 1 milliliter per liter. So we have set our micropipettes to 250 microliters, which is 1/4 of a liter. So let's just add that to the media. All right, next we are going to turn the magnetic stirrer off, and let's going to add the rest of our water. So for these media bottles, it does not say what is 250 milliliters, but
Yeah.
this edge right here, that's the 250 milliliter mark.
Oh, okay. Good
So
to know.
up next, we are going to calibrate the pH. So we have our pH meters, and we want to calibrate the pH. Actually, I don't know if that pH meter fits in that
Yeah,
bottle.
I was curious about that too. It looks like
Oh,
it.
just barely. Okay, perfect.
All right.
All right, we're looking for a pH between 5.4 and 5.8. So we'll just keep it in the middle around 5.6. Carnivorous plant, like I say, they like acidic soils, so let's just keep it a little bit on the acidic side at 5.6. When working with house plants, I usually keep it a little higher, around 5.8. So, but for carnivorous plants, let's do 5.6. So, I'm going to do one drop at a time. All right, we have calibrated the pH. Now the only thing left that we need to add is our gelling agent. So we're going to be using agar on this case. So we are going to add the equivalent of 8 grams per liter. So because we're making 250 milliliters, that means we need to add 2 grams of agar. So I have my 2 grams of agar, and I'm going to pour that directly in my media bottle.
Could you use gel and gum too, or for the Petri dishes, is it better to use agar?
Yes, so we are going to be sterilizing seeds. So one thing about the seed sterilization method that we're going to be using will be with hydrogen peroxide. And one thing with hydrogen peroxide and gel and gum, we have the residue of hydrogen peroxide in the media, which we will, because we're going to transfer the plants directly from the hydrogen peroxide to the media. The hydrogen peroxide can precipitate the gel and gum.
Oh.
So it will turn the gel back into liquid. This will be one of those special cases where we will have to use agar instead of gel and gum, because it will precipitate the media. And also one thing about gel and gum is that gel and gum does require calcium to solidify. We have 1/4 strength MS, so that means we have a reduced amount of calcium, which can make the gel and gum not solidify or maybe be softer than it would normally be. One case, you still want to use gel and gum, because I love to use gel and gum on my carnivorous plants. Like on her channel, we made the media for the carnivorous plant. We use 1/3 MS, and I still use gel and gum because I love it. It's like a really crystal clear. But when we go all the way to like down to 1/4, I prefer to use the agar, because I know it's going to solidify perfectly.
Gotcha.
Okay. All right. So our media is done. All we have left to do is close it, make sure it's not tight, and we're going to place this in the pressure cooker to sterilize for 25 minutes at 250 degrees Fahrenheit or 121 degrees Celsius or 15 psi. All right, we are back. We have the media is sterilizing at the moment in the pressure cooker. In the meantime, we are going to take our Explant for our Nepenthes. So this, if you are plant fanatic, this is going to be painful.
Look away.
This is going to be painful because we're going to take the node from the plant, and basically that means that we had to cut it. So, like I said, this is going to be a little bit painful, but I mean that's what we had to do for science, right? And we do this for you guys because you have requested to do Nepenthes for many years, and it's like I have my plants and I don't want to cut it, but it's like, well, I just got one more plant, and we are going to cut it and sacrifice it for science. So let's get started. So the first thing is that we need to make our sterilizing solution. So if you already watched Lauren's channel, that you saw that we did a sterilizing solution. We are going to use the same sterilizing solution, which it won't be bleach. Well, we're using chlorine, but we are not using the 10% or like your regular bottle of bleach. The reason we are not going to use the regular bleach is because these plants are very, very difficult to sterilize. Whoever has worked with carnivorous plants, you understand that Nepenthes, Venus flytraps, Sarracenias, Helianthura, Sondius, Pinguiculas, all of those carnivorous plants are very difficult to sterilize. They are very easy to keep in tissue culture after you successfully introduce them, but the first step to introduce the plants in tissue culture is super, super difficult. So the one way to get have a better chance of success is using a milder sterilizing solution instead of like your regular like 10% bleach. We are going to be using this. So this is you use this on your hot tub. So this is chlorine like sanitizer, like I say, for your hot tub. This is sodium dichloro. I will not say the rest of the name because I don't know how to say that, but we are going to have the name of the solution here. And just let me give you a close-up, just so you want to purchase the exact one that I have here on my hand, you can do it. This is, how much is this? This is like 2 pounds.
We'll make like tons and tons
Yeah,
of
this is
sterilizing solution.
2.25 pounds, which this is going to be a lifetime or sterilizing solution that you have, because this makes a low sterilizing solution. So on her channel, we already make the sterilizing solution for the Venus flytrap. We have a lot extra, so we are going to use this. So I'm not going to make more, but let me tell you how to make it, because it's very easy. You take 1 liter of water. This is just the still water. 1 liter of water. Then you measure. You want to do 250 ppms. I'm talking about parts per million. No, I'm not talking about plant preservative mixture. That's something else. But I'm talking about parts per million of this solution. So 1 gram of these pellets gives me 500 ppm. So I need 250. So that means I take half a gram for 1 liter of water. Then that's it. You add your dichloro in the water, get it in solution. So you mix it up until it dissolves, and then at the end you add this is polysolvent 20 or like twin 20. It's a surfactant. This is for the sterilizing solution. It sticks better to the tissue, to the plant material. So you have it does a better job sterilizing the plant material, the tissue. If you don't have a polysolvent 20 or twin 20, you can use a dish detergent. It works the same way. The only difference is that if you want to end up with a low foam, a low soap, that compared to something like a polysolvent 20, but it still works the same way. It's just a surfactant. It doesn't add anything else. So let's go ahead and take the Explant. I know I was trying to make this a little bit longer because I'm just trying to prepare myself for taking the Explant on this plant. But I think it's time. So what I'm going to do is going to take my little scissors. I'm going to clean them using some isopropyl alcohol. I'm going to take a paper towel. So I dry this up. You can also use 10% bleach. I'm not that worried about the virus from these plants. If I'm working with some other more virus susceptible plants, then I would use bleach to clean my tools instead of 70% alcohol, but I'm not that worried. You can use either both. You can also use heat. So if you have a Gladby sterilizer, you can also sterilize it with your Gladby sterilizer or with like a lighter, or just to heat it up, just so make sure they are nice, clean, and sterile. And now I want to cut this plant off. So I want to take the first cut, and I want to let Lauren take the second one. So I just want to cut. I hope I have a good angle right here, and I think the scissors maybe just a little bit
super
small.
tiny scissors.
I think we're fine.
Yeah. Trigger warning, plant violence.
There we go. So I just took my note. I hope the camera picks it up, but the note is right here. So I don't need this lip. So I just want to cut it away. So the node will be here in the middle. So the new plant, there will be one plant coming from here, and the other one I can see the node. I can see a little bit slightly shade, and
bump.
you can see a little bump right there. That's going to be the node coming from this other lip. So I have 2 nodes on here. I want to let you take the next one. So you can take this, and then I will say that you can either cut like 2 lips, so you have 1 lip here and 1 lip here, so you can just cut underneath there, or you can go ahead and take the third one if you want to, but basically all you need is use 1.
Gotcha.
But you can take 2.
Under this leaf here. It's the world's tiniest pair of scissors here.
I know, I should have grabbed the larger one. So for the other one, I just want to take the other lip as well.
There we go.
And I don't need the new lip. I took the top cut, So I don't need all of this because my meristem is in the node. So I just want to trim the end. And for the edges, because I cut this with the scissors, I still have these edges. So I want to do this with my hands carefully, just try to remove some of this material. You can do this with the scissors as well, but I found that using my hands, I do a little bit better job. There we go. So one of the hardest things to sterilize about the Nepenthes and the way it grows, because it grows one leaf on one side and then the other side, is this part. So right here, you see that there is a leaf that is covering. So there is you can see a little empty space right here. So trying to get the sterilizing solution there, that's usually where all the contaminants will come from, right here. Because the bleach may not be able to get in there, and it won't be able to sterilize the plant.
Yeah.
So that's where I want to come over with my fingers and try to peel that up until I don't have that gap in there. So this, it's a little bit patient, but I found that, like I say, it's easier to do this by hand.
Should I take this rest of the leaf off here?
Yeah, you can take that off, so you can do it with your hands,
do it
or you
with
can
my
do it
scissors.
with the scissors. I think I took your scissors.
It's okay.
Yeah. Perfect. So this will be difficult to see in camera. Actually, let me get close to the camera so I can show this better. Okay, so right here on this edge, I see dirt, which is not great. So that's where I say that this is, if I can get the camera to focus, there we go. So there is a little bit of dirt right there. So this is the hardest part to sterilize. So just try to remove as much as you can, but make sure not to get all the way to the meristem, which is at the very, very tip. So on the other side, you see that I cut it up. So I clean this a little bit better, but there may be still a bit of soil in there. So that is the reason why we use dichloro, because we use a much longer sterilization time.
Did I take too much of this node off of this guy? I feel like mine.
No, I think looks fine. Let me see. Let me inspect it. Oh.
Sorry.
No, it's fine because you see the note in here.
Oh.
I want to show it to her, and then I show the camera. The note is right there. You see that little dot right there?
I see it.
That's your meristem.
I almost chopped it.
Yeah. So basically, there are some other methods. With this plant, I won't be able to. It won't allow me. This plant won't allow me to show you the other methods. But if you remember in class,
The Monstera.
the Monstera, there is a very good example where if you don't want to chop the entire plant, you can just trim with your knife, with your scalpel, trim the part where the meristem is. So, and you still don't have to cut the plant like we did. But with this plant, it is so small that it's very difficult to do. So it was better just to cut the entire node. And also when you have the entire node, the plant grows much faster. The only problem is that you have a bigger chances of contamination. But this one looks good. Let me just show the camera pretty quick. All right, so what I was showing Lauren is like this little tip right here. This is your meristem, so this is where the plant will grow up. So it was very close. See, she just cut it right here, but it will be okay because this is where the new plant will grow from. And you have two nodes or one? You only took one, right?
Yeah,
From one leaf?
I just took one.
Oh, you just took one leaf.
Yeah.
Okay, perfect. So right there, I took three. I have 3 nodes in mine. So on mine, I will cut mine in half.
To get more explants.
So I have 2 explants. Yeah, there we go. So I'll cut mine in half. I have my jar over here. So I just want to put my 2 nodes in my jar. I actually want to save these leaves because one other experiment, make sure to check the channel because I want to do an experiment on trying to regenerate plants from Callus. So I want to use the leaves to try to regenerate Callus, and then later on I see if I can get plants from here. But if this is something I haven't been able to, in theory that it is possible that you can take regenerate plants from any part of the plant. but I haven't seen this done. So make sure to subscribe to the channel because there's something that will be coming hopefully soon where hopefully I can get Callus from these leaves. So I want to save these leaves for my next video.
Someone was asking me on Instagram about growing Monstera Thai constellations from leaf cuttings, and I was like, I wouldn't, that would just take forever, I feel like.
It's not ideal to grow plants from Callus, but if it's something like this where you don't want to sacrifice it, that will be the way to do it. But it's not really worth it. Sometimes it's faster to let the plant grow, take the explants from there than trying to grow from Callus.
Yeah.
But people have been asking, so and it will be a nice experiment to see if we can get that done.
Cool.
So let's get the sodium dichloro, and I just add this to my jar. I want to close the jar, and always when I close the jar, make sure to also flip it a few times, so you are also sterilizing the part of the cap and everything that is not in contact with the dichloro. So I just flip it upside down, just give it just shake it for just a few seconds, and this we are going to sterilize the plant for 24 hours. So they are going to be sitting in an orbital shaker, and they're just going to do this for 24 hours. So we are going to come back tomorrow, and we're going to place the plants in the media that we already made. All right, so if you thought we were done with the Nepenthes, you were wrong, because we are now going to be sterilizing some seeds. That's also something that people have been asking me for a very long time on how to sterilize Nepenthes. So I just got a few Nepenthes from a friend. So I want to do the we're going to be doing the sterilizing solution. Very similar to other methods, like other videos that I showed that I've been using the hydrogen peroxide. This is 3% hydrogen peroxide from the grocery store. Works great for all of the other seeds that I've been testing. For pretty much all the seeds I've been testing. So I want to test it out with the Nepenthes. Sometimes now when working with Nepenthes, you need to work with very fresh seeds. They have a very short shelf life. Hopefully this is still fresh enough for me to do it, but that's another reason why I like to use the hydrogen peroxide, because hydrogen peroxide adds oxygen to the seeds, which is essential for the seeds to sprout. So we are going to be taking 1 milliliter of hydrogen peroxide. There we go. So 1 milliliter of hydrogen peroxide. So one for me and one for you. And we have the water. We're going to take 19 milliliters of water. So we are going to make a 20 milliliter solution. 19, 20, let's take a look. Just a bit more. Perfect.
You're very exact. I'm just like, whatever, it's close enough.
Perfect, 20 milliliters. So while she does that, I want to take a few of my seeds and I'm going to place this in my test tube. So I'm going to take some of my seeds and I'm going to place those in my tube. Some people like to use filter paper or like coffee filter paper to place the seeds, and then to make like a little envelope, because these seeds are really small, but I just want to use my tube.
I was talking to Justin from Created in a Garden. He told me he uses origami paper.
Origami
like black
paper.
origami paper.
Black origami paper.
So you can really see
Oh, so you can see the seeds.
Yeah.
Yeah, good idea. And I guess if you're working with like black or dark seeds, you can use the coffee paper.
Yeah.
Yeah, or filter paper. So I have my seeds, and I want to add my my solution in here. I typically don't add 20, but I will add just a drop just to have a little bit better sterilization effect to it. So just one drop for you. And if you are working with really old seeds, if you're working with Nepenthes, old seeds, they may not sprout, they may not be viable, but you can also add a little bit of sugar to your hydrogen peroxide, because sugar is it gives them the energy. Sugar is energy for the plant, so that gives the energy for them to be able to sprout, because these seeds are tiny. These are really, really small seeds, so they don't have a lot of starch. Like you take, for example, corn. Corn has all the white or yellow part of the seeds or the kernel. It's a starch, which is a type of sugar, which that's energy for the seeds to sprout. But these seeds are really small. They don't have that much energy to be able to sprout. So you can add a little bit of sugar to this solution to help them to sprout as well. So let's just take our hydrogen peroxide solution and fill it pretty much all the way to the top and put the cap on.
I wasn't expecting them to float up to the top.
I probably added a little bit too much, but I think that'll be okay.
There we go.
There we go. So there we go. They are ready. So we are just going to leave them overnight. Same thing as the Nepenthes cuttings and the Venus flytraps. The hydrogen peroxide will, in the light, will, but tomorrow it's just going to be water. It's going to be sterile water. So that's another reason why I love to use hydrogen peroxide to sterilize my seeds, because I don't have to rinse them. With bleach, I had to rinse them, but with hydrogen peroxide, I don't have to. So these seeds are ready, and we come back tomorrow. Okay, so our media is done. I'm going to pour it in some Petri dishes. So I'm spraying all my surfaces. Make sure everything is clean and sterile, no contaminants. I also spray in my media. So I want to pour one bottle in one sleeve of Petri dishes, and I want to let Lauren do her media bottle as well. So all my surfaces are clean. My media is clean. First, I want to take the Petri dishes out of the bag. I also want to spray the scissors that I want to use to open the bag. But to put it upside down. I want to take my bag and move it to the side, just out of the way. Make sure to keep it closed. Do not contaminate the bag, because we are going to use it to put the Petri dishes back in. And I just want to open my Petri dishes. And we are going to pour this by hand. So I'm using the paper towels just so I don't burn my hands, because the media is still pretty hot. For my media, I want to let it solidify like this. So it's going to take like 10 minutes before I put the lid back on and I close and I place those back in the bag. So we just wait 10 minutes and we'll be right back. It has been 10 minutes since I poured the media. So I just want to spray my hand with isopropyl alcohol. Then put the lids back on. Stack them. Like this. I take my bag, flip it. I know the media is upside down, that's okay. And just going to close this with my tape. So our media is ready for us to use tomorrow. All right, guys, so we're back 24 hours later. We have our Nepenthes cultures right here. They are doing great. You can see some of the sterilization has damaged some of the tissue, but that's exactly what we need. We need that's exactly what we need. We can see that it is working. All we have to do now is trim it. So I'm here on the laminar flow hood, and the first thing I want to do is spray my glass jar before I place it in the hood. I also have a sterile water that I made. So this is just the still autoclave water. So sterile water used to rinse the plants. I will spray my jar in here, and I want to trim the plants, rinse the plants and trim them. I want to do it different that you may used to, because what you're used to is go from the bleach to sterile water and then trim. But I want to do it a different way. I want to take some of my dichloro, place it on my plate. That's going to help me keep a sterile surface. And I also taking my notes right there in the dichloro. Take my scalpel, and I want to trim it in here. So I need to trim the bottom, just slightly. I don't need to take a lot. I just need to take the very edge, and I know my meristem is inside there, so I don't need this part. And the top, I also want to trim that just a little bit. Just like that. That's just, so that is enough. So on this one I have two nodes. One is still in here, and the other one is right where that leaf used to be in there. So that is one. We'll do the same one to the second one. Trim the top. Trim the bottom. And the part of the leaf. There we go. So I have trimmed my plants. Now I will open my sterile water. Now, because this is a very mild solution of dichloro, it doesn't take that long. Basically, it's just a quick rinse. So I just dropping my notes in there, putting the lid back on, the cap back on. Just give it a quick rinse, just for a minute or two, and then I'm going to place this in my media. This is the media that we made yesterday on Laur's channel. We made it using gel and gum. You look how clean, look how clear this is. This is the Plant Cell Technology gel and gum, crystal clear. And normally, if I'm starting cultures, I start them on test tubes or smaller containers. But we made the media on the big round containers, but that's still okay. The only thing is that if one gets contaminated, then you lose the entire. These contain more media, so it can be a little bit more wasteful if one gets contaminated. If you have a test tube, you only use a very small amount of media, so it is. You don't waste as much media. So this is one plant. Then the second one. Take my node and make sure I want to stab it right there in the media, right in the center. Make sure it's facing up. And I don't want to go too deep, just get it just under the media, right there. So that is perfect.
Looks good.
All right, so now we want to have a, all right, we want to have Lauren do her plant.
All right.
Okay, we have Lauren taking the plant from the media, so she's going to take it out of the dichloro, place it on the Petri dish, and you can pour some dichloro in there if you want to.
Oh. And all over the table.
A little bit on the table is fine.
My bad. All right. And then we cut. Why do you pour the dichloro onto the plant?
So because I like to use fire to sterilize my plate,
Yeah.
but because I'm not using that, I'm not sure if it's 100% clean just by using the alcohol. So I like to spray the dichloro on the plate, so it's still on a sterile clean surface.
Shake it up.
Shake.
All right. I'll take her out. She's evading me. There we go. I'm gonna try to do it right way up.
Right. And just goes in the middle. Perfect.
Perfect.
Excellent.
There she is.
Excellent. What I'm doing right now is moving the seeds out of the hydrogen peroxide and placing them in the media we made for the seeds. Remember that this media is 1/4 the strength of full MS. We also have 30 grams of sugar and no hormones. For the leaves, I'm trimming the edges so I remove all of the dead plant tissue, and I'm placing them in media for Callus. So this media, I will leave this on the screen, is the exact same media, however it does contain hormones which are TDC and NAA. Alright guys, I hope you have enjoyed today's video. Thanks Lauren for being here with us today. We have done 2 videos, so we have done Nepenthes here on this channel, and we have done Venus flytraps on Lauren's channel, so make sure to check Plants in Jars YouTube channel. And if you need anything else for your plant tissue culture needs, make sure to check plantcelltechnology.com. And thank you and goodbye.
Bye guys.
Bye. Thanks for watching the video. If you are enjoying these tissue culture videos, make sure to follow us on social media for more informative content. And if you are interested on conducting your own tissue culture experiments, make sure to check us out at plantcelltechnology.com for all the products you need to get started. Use code FP10 for 10% off on your first purchase.